A conclusion to this experiment. Ionic compounds dissociate when put in an aqueous solution. We observe this when NaCl dissociated into Na plus sodium and chloride Cl minus. This allowed for Cl or chlorine to diffuse. Sodium sulfate dissociated to sodium sulfate ions. Note here that the bond in between this sulfur and the 4 oxygens, they're covalent. Ionic compounds dissociate when put into an aqueous solution. These are covalent bonds. Here, there's another thing I want to point out is that in this experiment, in the extracellular space, we had NaCl, which contains sodium. And in the intracellular space, we had sodium. So, this is why we didn't really test for the diffusion of sodium. It was already present in both. So, if we did test, we're going to find out that it was present in both. Now, this may be because it did diffuse, or it didn't diffuse. We would have no idea. We learned that certain molecules are too large to passively diffuse across a membrane. Again, in this lab, we're viewing size, and protein, such as albumin, and carbohydrates, such as starch, were too large to diffuse through the tiny pores in the dialysis tubing. These molecules would require facilitated diffusion, such as transport proteins, which are not available in dialysis tubing. Many ions can passively diffuse across a membrane. However, these diffusions occur at different rates, and molecular weight was a factor in this rate. So if you remember, 96 molecular weight of sulfate, 35 molecular weight of chloride. This 35 AMU, the rate of diffusion in this chloride was high enough to reach equilibrium, while the 96 was not, so the diffusions occur at different rates. Here's just a pretty little chart showing our results, and I'll again recap what they mean. We tested the starch and sodium sulfate solution, which was inside of the cell, the albumin solution, which was extracellular. This also includes NaCl. Can't forget NaCl, it's important. But we tested tube 1 and tube 5 for starch. Positive here, negative here. Starch did not diffuse across. Starch could not go from here to here. Sulfate, however, tube 2 and tube 6, both are positive. So sulfate did diffuse across. Let's imagine this line here is a selectively permeable membrane. So, and SO4, that's horrible, diffused across this selectively permeable membrane, which in this experiment was only factored by size. The chloride ion test, tube 3, tube 7, both are positive. Chloride ion started here, it diffused and reached an equilibrium. Chloride ions reached an equilibrium over here, 2 over here, and 2 over here, equilibrium. The chloride ions diffused and they reach an equilibrium based on the positive and similar amounts of the precipitate. Lastly, protein test, tube 4 and tube 8. Light blue over here, no protein in the cell in the beginning, but there is albumin right here, protein, and that's why this test was positive. And we can say that albumin did not diffuse because of its large size, hence the negative test over here. And that is this is experiment E. Yeah, experiment E, lab 6. Thanks.